Principle and Protocol of the Fast Chromatin Immunoprecipitation Assay. Produced by Creative Biomart. Part 1, Introduction of Chromatin Immunoprecipitation. Chromatin immunoprecipitation, also known as CHIP, is a type of immunoprecipitation experimental technique, used to investigate the interaction between proteins and DNA in the cell. The function of the assay is that it gives a researcher the ability to not only detect a specific protein-DNA interaction in vivo, but also determine the relative density of factors along genes or the entire genome. Part 2, Traditional and Fast CHIP Assay. Though powerful, the traditional CHIP assay is time-consuming and laborious. The fast CHIP assay can greatly reduce the time and labor involved. The improved assay is especially useful for studies which involve many samples, including the probing of multiple chromatin factors simultaneously, and looking at genomic events over several time points. The slowest step of the traditional CHIP assay is the 5-hour reversal of cross-linking, and the most laborious step is the DNA cleanup, which involves phenylchloroform extractions and ethanol precipitation. In fast CHIP, cross-links are reversed during a 10 minutes incubation, at 100 Celsius in the presence of Chelix 100. In addition, since fast CHIP does not require the addition of sodium bicarbonate or SDS buffer to elute the chromatin from the beads, the high temperature is sufficient, the DNA cleanup step is not necessary. After the 100 Celsius incubation, the DNA-containing supernatant is directly used in PCR. Another improvement in fast CHIP is the use of an ultrasonic bath, to increase the rate of antibody-chromatin interaction. In the traditional CHIP assay, the antibody-chromatin incubation can take anywhere from 1 hour to overnight. With the use of the ultrasonic bath, this incubation is decreased to 15 minutes. Part 3, Workflow of the Fast CHIP Assay. The CHIP assay begins with the cross-linking of protein, DNA complexes, by the fixation of cells or tissues with formaldehyde. After lysing the cells, the nuclei are disrupted, and the chromatin is sheared either by sonication, or by digestion with micrococcal nuclease. The chromatin fragments, typically between 500 and 1000 base pairs in length, are immunoprecipitated using an antibody specific to the protein of interest. Part 4, Materials of the Fast CHIP Assay. Reagents include proteinase FROs, phosphate buffered saline, SYBR green PCR master mix. Equipment includes sonicator, refrigerated microcentrifuge, heat blocks and hot plate, tube rotator or tumbler, quantitative PCR machine, and ultrasonic bath. Buffers and solutions include glycine, IP buffer, lysis or sonication buffer, Chelix 100, Proteinase K, and TE. Part 5, Protocol of the Fast CHIP Assay. First step, cross-linking. Firstly, approximately 400,000 to 4 million cells are required per IP sample. Secondly, add formaldehyde to the tissue culture medium directly, swirl the dish and incubate for 15 minutes. Thirdly, quench formaldehyde by adding glycine and incubate for 5 minutes. Fourthly, harvest cells by scraping and centrifuging. Fifthly, keep cells on ice and wash twice with ice-cold IP buffer. Second step, sonication. Firstly, lyse approximately 10 million cells by resuspending in ice-cold lysis, and pipetting up and down several times. Secondly, collect the insoluble material by centrifuging. Thirdly, repeat the step 1 and 2 once more, collect the pellet by centrifugation and aspirate the supernatant. Fourthly, resuspend the pellet in ice-cold lysis, and split into 2 fractions. Fifthly, the protocol used for sonication can vary widely, and must be optimized for each cell or tissue type and sonicator setup. Sixthly, after sonication, The chromatin should be cleared by centrifugation. Seventhly, transfer the supernatant to a new tube and aliquot for storage. Save one aliquot for extracting total DNA for the input sample. Third step, isolating total DNA. Firstly, precipitate DNA from the aliquot for 10 minutes with absolute or 96% ethanol. Secondly, pellet the DNA by centrifugation. Thirdly, aspirate or decant the supernatant, and add 75% ethanol. Fourthly, centrifuge, and remove as much of the supernatant as possible. Fifthly, dry the pellets to completion. Sixthly, add Chelex 100 slurry to the dried pellets. Seventhly, boil for 10 minutes and cool by centrifuging. Eighthly, add Proteinase K to each tube and vortex. Briefly centrifuge to bring contents to the bottom of the tube. Ninthly, incubate at 55 Celsius for 30 minutes, gently resuspending the Chelex once or twice during the incubation. Tenthly, boil for 10 minutes and centrifuge the condensate to the bottom of the tube. 11th, transfer the supernatant to a new tube. 12th, add DD water to each tube containing Chelex slurry, vortex, and centrifuge the contents to the bottom of the tube. 13th, remove the supernatant and pool with the supernatant from step 10. 14th, the DNA can be run undiluted on agarose. Fourth step, immunoprecipitation. Firstly, for each IP sample, dilute the equivalent of 1 million cells of chromatin with ice-cold lysis. Secondly, add specific or mock antibodies to each sample and mix by inverting. Thirdly, float samples in the ultrasonic bath for 15 minutes at 4 Celsius. Fourthly, clear the solution by centrifugation. Fifthly, while the chromatin and antibodies are incubating, transfer protein agarose slurry to a clean tube. Wash 1 to 3 times with IP buffer to remove ethanol. Sixthly, resuspend beads in IP buffer. Dispense the diluted slurry to new tubes, 1 tube for each IP sample. Centrifuge and aspirate buffer. Visually inspect tubes to make sure each one has the same amount of beads. Seventhly, transfer no more than the top 90% of each cleared chromatin sample from step 4, to the tubes with the beads. Eighthly, rotate tubes with a rotating platform or tumbler. The rotation should be fast enough to keep the beads suspended. Ninthly, centrifuge the tubes and aspirate the supernatant. Tenthly, Washington the beads 5 times with ice-cold IP buffer. After the last wash, remove as much supernatant as possible without removing the beads. 11th, add Chelex 100 slurry to the washed beads. 12th, add Proteinase K to each tube and vortex. Briefly centrifuge contents to the bottom of the tube. 13th, incubate at 55 Celsius for 30 minutes. Gently resuspend beads and Chelex 100 once or twice during the incubation. 14th, boil samples for 10 minutes. 15th, centrifuge to cool samples and bring condensate to the bottom of the tube. 16th, transfer supernatant to new tubes. 17th, add DD water to each tube containing Chelex or protein beads slurry, vortex, and centrifuge contents to the bottom of the tube. 18th, remove the supernatant and pool with the supernatant from step 16. 19th, the PCR-ready DNA can be stored and repeatedly thawed and frozen over several months, without loss of PCR signal. Fifth step, analysis. After reversing the crosslinks, the DNA is isolated, and used in one of the several detection methods including PCR, qPCR, sequenced using a rapid sequencing technology, or hybridization to a DNA microarray. Enrichment of a particular DNA region over other sites where the factor is not expected to bind indicates that the protein interacts with this region. Part 6, Notes of the Fast CHIP assay. Firstly, The cross-linking times and formaldehyde concentrations used here are suggestions, and may need to be optimized depending on the cell or tissue type used, as well as on the factor being immunoprecipitated. Secondly, both PMSF and lipeptin have short half-lives in aqueous solutions at room temperature. It is important to prepare the lysis or sonication buffer fresh, and keep it on ice before use. Thirdly, the chromatin preparations can be stored at -80°C for months without loss of pull-down efficiency. However, repeated thawing and freezing can reduce this efficiency. Fourthly, to reduce nonspecific binding to the protein beads, blocking reagents may be used to block the beads, both prior to the IP and during the IP. Fifthly, for some antibodies the amount required may need to be determined empirically. Sixthly, if an ultrasonic bath is not available, samples may need to be incubated for 1 to 2 hours at 4°C depending on the antibody. Seventhly, nonspecific binding of the chromatin to the protein beads accounts for the majority of the Mach signal. Therefore, reducing the amount of beads used may reduce the Mach signal. If you want to get more information, or you have any questions about experimental protocols, and other biological knowledges, please contact us via email. You can also log in our website. You can find me at www.creativebiomart.net for more information. Thank you for your attention.